In 1865, by means of his pea plant experiments, Gregor Mendel found out that each characteristic in an organism is controlled by a specific gene. In 1874, Friedrich Michner identified the DNA in a cell. In the 1920s, scientists believed that the genes consist only of proteins. In 1928, Friedrich Griffith determines that genes consist of DNA and not proteins. By 1944, Oswald Avery supported Griffith's theory that DNA is the hereditary molecule. By the 1949s, Edwin Chargaff determined that there are equal numbers of adenine and thymine bases, as well as equal numbers of guanine and cytosine bases in a DNA molecule. Then along came Rosalind Franklin. In 1952, she took an X-ray photo of DNA, and Maurice Wilkins showed this photo to Watson and Crick without Rosalind Franklin's permission. By 1953, James Watson and Francis Crick formulated the double helix structure of the DNA using a three-dimensional model. This is a well-known model that we still use today. In 1962, Watson, Crick and Wilkins received the Nobel Prize for Physiology/Medicine. In the 1990s, the Human Genome Project was established with James Watson as the first director, and by 2003, the entire human genome was mapped. DNA is found in the nucleus of a cell. Each nucleus is surrounded by a double nuclear membrane, which is perforated by pores. It's these tiny holes. It contains nucleoplasm, a nucleolus, and a chromatin network or some chromosomes. Chromosomes contains the genetic material of a cell. The nucleus was the first organelle within a cell to be discovered. It was described by Franz Bauer in 1804 and in more detail by Robert Brown in 1831. The nucleus controls the structure of a cell. It's involved in the production of ribosomes, RNA, and substances needed for the synthesis of proteins. Nucleic acids are the largest molecules found in living organisms today. They were discovered in the late 19th century as acid substances associated with the proteins of the cell nucleus, hence the term nucleic acids. Chromosomes are thread-like structures located inside the nucleus of cells. A karyotype is an individual's collection of chromosomes. A normal human karyotype consists of 22 pairs of autosomes and 2 sex chromosomes. Imagine this karyotype as 2 sets of encyclopedias coding for all the information in your body. One set came from the sperm cell and the second set came from the egg cell during fertilization. So the chromosomes are the structures within the cells that contains a person's genes. Genes are segments of DNA that carries the code for a specific protein that functions in one or more types of cells in the body. There are variations of these genes known as alleles. Homologous chromosomes are made up of chromosome pairs of approximately the same length, centromere position, and staining pattern for the genes with the same corresponding locus. DNA is the smallest part that, together with proteins called histones, forms a chromosome. A chromosome is therefore nothing but a chain of DNA that has been made compact enough to fit into a cell. Each chromosome is made up of DNA tightly coiled many times around proteins called histones that support its structure. Chromatin is a complex of macromolecules composed of DNA, RNA, and proteins, which is found inside the nucleus of eukaryotic cells. The primary protein components of chromatin are histones that help to organize DNA into bead-like structures called nucleosomes by providing a base on which the DNA can be wrapped around. DNA is a molecule composed of two polynucleotide chains that coil around each other to form a double helix carrying genetic instructions for the development, functioning, growth and reproduction of all known organisms. Adenine is a molecule with the chemical formula C5H5N5. It's one of the two nucleobases used in forming nucleotides of the nucleic acids. Guanine is C5H5N5O. It's a purine base that is a constituent of nucleotides occurring in nucleic acids. Cytosine, with its formula C4H5N3O, is a pyrimidine base found in DNA and RNA that pairs with guanine. Thymine, with its formula of C5H6N2O2, is a pyrimidine nucleobase that is uracil, in which the hydrogen at position 5 is replaced with a methyl group. Phosphoric acid is the second constituent of the nucleic acid. These molecules form the phosphate group of the complex nucleic acid molecule. Pentose sugars are monosaccharides or single unit sugars containing 5 carbon atoms. In a solution, there is a rearrangement of the bonds and they form the characteristic pentagonal or ring molecule. Deoxyribose is so called because it has one less oxygen atom than ribose. Nucleotide consists of a pentose sugar, either deoxyribose in the case of DNA or ribose in the case of RNA. It also contains a phosphate group and one of 5 possible nitrogenous bases. Complementarity is achieved by distinct interactions between nucleobases, adenine with thymine and guanine with cytosine. In a nucleic acid, the nucleobases are held together by hydrogen bonding, which only works efficiently between a purine and a pyrimidine. Therefore, adenine binds to thymine and guanine binds to cytosine. The sugar-phosphate backbone forms the structural framework of nucleic acids, including DNA and RNA. This backbone is composed of alternating sugar and phosphate groups and defines directionality of the molecule. The sugar is at the 3-accent end and the phosphate is always at the 5-accent end of each nucleotide. So as we've mentioned, DNA consists of a double strand. The one strand acts as the template strand. It refers to the sequence of DNA that is copied during the synthesis of mRNA. The opposite strand is called the complementary strand because it complements the template strand. The smallest genes are for tRNAs, and they're about 76 nucleotides long. The longest gene is perhaps the human CNTNAP2, which is just over 2.3 million bases long. When you read a sentence, the meaning is not just the sequence of the letters, it's also in the words those letters make and in the grammar of the language. In the same way, the human genome is far more than just its sequence of letters. To most, it is a foreign language, written without capitalization, punctuation, without any breaks between words, sentences or paragraphs, and with some additional letters placed in between that doesn't make sense to us. Scientists can't even agree on the number of genes and proteins available in the human body. So there is still a lot of work that needs to be done until we fully understand the human genome. If you stretched out or laid together the DNA in a single cell in the human body, it would be approximately 2 meters long. This DNA needs to be coiled into chromosomes to fit into the cell. In our next edition, we will look how these genes codes for specific proteins and how the body decodes this message. The FOSS education videos aim to make content colorful and clear to understand. The videos are based on the CAP syllabus in South Africa. To learn more, you can watch some of our other videos, which includes the introduction to genetics, DNA structure,